The gut microbiota consists of many different types of microorganisms, including bacteria, archaea, and fungi. So generally, many people focus on just the bacterial portion. What we did is we were able to account for the fungi and archaea as well within 9,600-ish different metagenomes. This allowed us to study previously unknown genes within the gut microbiota, which we know should be associated with health and disease. By looking at these genes across so many samples, we were able to make associations between the genes, their functionality, and the different populations they occur in.
We looked at metagenomic samples in a lineage-specific way. So instead of just looking at bacteria, we looked at whether the specific samples came from bacteria, archaea, viruses, or eukaryotes. And then depending on the origin of specific genetic material, we applied different gene prediction tools. We also looked at specific codon tables, so the groups of 3 DNA bases that correspond to a single amino acid. This is also different depending on which organism a sequence comes from, and our research looks at this in a different way from the majority of other papers. The metadata that we looked at included not just data about the gender, the country of origin, or the age of patients, but also the disease data. So we can potentially link specific genome metagenomic variations to diseases in the future, and also potentially whether there are antimicrobial peptides in these metagenomic sequences. We clustered over 8 million protein clusters, and of these there were 69 small protein clusters present in over 90% of patients, of which around 1/3 had antimicrobial activity in silico, according to some software analysis. So far, we've only looked at metagenomes in human stool samples, but what we would like to do is also incorporate additional samples coming from the small intestine, the oral microbiome, as well as environmental samples such as forest microbiomes and marine microbiomes. Then not just looking at the metagenomes, but also RNA and proteins and metabolites, and how bacteria flow from one environment to another in this concept called One Health, with gene flows back and forth between humans and the environment.